Good morning, Gavin Lopez, the most beautiful child in the room. Good morning, Abuelito. Abuelito. Singing songs about Abel. Abuelito. Hello, Diego. Go, Diego, go. You guys that are here, go ahead and go to Schoology. Go to today's lesson, testing selection. Testing selection. We're actually going to be here for 2 days, but go to this location. When you get there, there are a couple of documents that we're going to use. One of them is going to be here, and another one is this AP Biolab. We're going to be using both of these. What I need to let me know if you're here, click on Artificial Selection Lab. To let me know that you're present today, go ahead and go to today's lesson and open Artificial Selection Lab. Good morning. Good morning. Let's see, they got people in the waiting room. Let's see if I can get all these guys in here. Quick, fast, in a hurry. Quick, fast, in a hurry. Okay, good guys. It is 8:30. Nobody else in the waiting room. Looks like a little over half of you guys have already made it. I obviously, for those of you guys that are in person, I'm not here, at least during first period. I have a doctor's appointment. I'm at the hospital. I have to go in and take a stress test at 9:30. So I have got to make haste. So I need you guys to work with me and go to today's lesson at 4, and it is 4:30. That would be April the 30th. And there's 2 documents that we're going to use. I have made one of those is going to be Artificial Selection Lab, and the other one is Artificial Selection Student. It is an AP lab. It is an AP lab. It's perfect for what we're doing today. What I do need you to do is click on this document, Artificial Selection Lab. That lets me know that you are here and you're hearing me. And then this is the one that the same document is right here, AP Biolab Artificial Selection. That's the one we're going to actually use. This lets me know that you're here. This is the one that we'll use. They're the exact copy, but this is just, you might want to even annotate on this one if you want to. That has the same information, but I think this one's going to read a little better for you. I also did not know whether your Chromebook could open a PDF, but this is the one that I'm going to use. So I'm going to click over there and let's see how many of you guys have made it so far. George has made it. Beltran has made it. LeVar has made it. Cuenca's making it. David's made it. Diego, we're waiting on you. Janaya, waiting. Gavin has made it. Thank you, Gavin. Thank you, Abuelito. Thank you, Dev. Thank you, Manas. Thank you, Ashmeet. Okay, enough of you guys have got it open. The one that we're going to use, however, is going to be, let me let these guys in. The one that we're going to use is going to be the PDF, the one I'm going to use. So if you can use that one, use that one. I'm going to go ahead and open it now. And I'm going to put on the timer. It's 8 pages, but they're not full on pages. They're not like, some of them have a lot of graphics. We're going to start with reading, and then we're going to discuss it. I'm going to ask you guys some questions. So just read through this document. I'll give you guys 10 minutes. Please make note, jot down like I have a question, and if you have a question, what page, what paragraph, what line. We have some, if you need to ask me some questions, I'll ask you guys some questions, and then we're going to set up, a lab. So I'll be quiet. I'll give you guys 10 minutes. Again, if you just got in, we're at April the 30th. We're at April the 30th. And we're open this lab and we're reading through it. I'm going to use this as a way to take attendance. If you've opened this document today, Artificial Selection Lab, I will count you present and just need you to open it so I know. And then here, this is the one that we'll actually read. They're the exact, it says this is the original copy. It's easier to read the PDF if you can open PDF. If you don't have a PDF, or if you can't open PDF, then open the Google Doc. Jacob, you just got here. The instructions are to open these 2 documents, but stay on this document, this PDF, AP Bio Lab Artificial Selection. Read that. We're going to come back together at 8:46. No, excuse me, 8:44, excuse me. Sarah, you need to open this document, both of these documents. This tells me that you're here. This is what we're going to be using. They're the same document, just one's a Google Doc. And just read it. Okay. Cuenca, we're reading these 2 documents. They're the same document. You open this one just to let me know that you're here. And then this is the one that you'll use. And we're quietly reading this till 8:44. When you're reading it, maybe jot down some ideas, some notes, some questions. Yes, Sarah. Yes, Sarah. There's two of them. The PDF is actually easier to read. I'm just taking attendance from you clicking this and open it. Some people may want to use the Google Doc to take notes. So I made that available as well. You got me, baby? You heard that? You heard me, Marino? Thank you, baby. All right, you guys be good. I'll be quiet. Let's see your attendance. Oh, shoot. I don't think so, Kimberly. No. I mean, similar insofar as they use light. All right, we're going to come together in 30 seconds. We're going to come together in 30 seconds. Please look at the screen that I am sharing. It is important that you see the screen that I'm going to share, or I am sharing. So if you look at our agenda today, we've got a couple of things. So the introduction to new material is that you guys are going to assist me in setting up a hypothetical experiment. You've just read through it, and then I might change a few things. My students did this 2 years ago, and one of the students used these for their IAs. They changed it and modified it a little bit for their IA. It's a pretty cool experiment how they altered it. But I realized from reading your proposals that you need to be more comfortable with experimentation, and it's hard to do that, right, when we're not actually doing it. So I'm going to drop you guys into a breakout room. I'm going to bust into those breakout rooms, and when I do, I not should hear people just typing back to one another in a chat box. That is super mediocre, and we don't have time for that kind of thing. So when I bust into that breakout room, I need to hear you guys discussing it, just kind of getting your ideas out there. What was this about? Was there something you thought was cool, something you didn't, you thought you were questioning? What is it that's going on here? And then I'm going to show you guys how to set up an experiment using the IVCDV method. There's a method for this, and this will make things more clear for you for your revisions. Your revisions are due just in a little over 2 weeks, a little under 2 weeks, excuse me. So I'm going to put you in breakout rooms, and let's see. Turner, when I go into those breakout rooms, what should I hear? Turner, don't chat to me, kid. Yeah, exactly. It's the voice not chat, and you're chatting with me. What is that? Yeah, and okay, when you go in there. All right, here we go. You guys have 5 minutes, and I'll start coming in about 30 seconds. Just what is this that we read? What are some points that you didn't understand? What do you think the independent variable is? What do you think the dependent variable is? Just talk. And you have 5 minutes.
Mr. Pope, it's not letting me join.
Expand your screen full screen. Put your screen on full screen. You should see it then. Do you see it now?
No, I see it's up there, but when I click the join button, it doesn't want to go through.
All right, hold on a second. I'm going to see if I can drop you into a different room. So let's go to try this one, see if it helps, see if that helps. Andy?
Mr. Pope, most of us in this group are actually in the classroom. So
yeah, so you can talk. Yeah, just talk to one another.
Yeah, but what we were wondering is if you could put us all in this group, and then
we're only going to be in this group for about 3 more minutes. So we're not going to spend much time here. But I will in the future do that. Yeah, sorry about that.
Yeah. Alrighty.
I don't want to interrupt somebody else's conversation. You weren't able to do it?
No, I wasn't able to. Do you just want me to leave and rejoin?
No, what I think you could do is, Turner, are you still here?
Yeah, I'm in breakout room 3.
Hey, why don't you just talk to Brianna, because she can't get in the room, and you guys just make a group. I'm going to jump into another group to check on them. Just stay here and talk to Brianna about it.
Okay.
Okay.
Hello.
The only question I had was that I don't know if I wasn't here for class that day, but does anyone know what a trichome or trichomes are?
Ooh.
I don't know if I was here for that class.
You weren't here. You weren't here.
I don't remember.
I'm kidding. I'm going to show you what a trichome is in a second.
Oh, okay.
I was like confused on that same part as well. Like finding.
Y'all got a bunch of questions?
Do we have questions, y'all?
I'm still trying to read through the whole thing.
Yeah, Sam, I didn't finish.
Well, I figured you guys wouldn't finish most of it, but I mean, excuse me, you'd finish most of it, but not all of it.
Yeah,
Just
yeah.
talk about what you already have read, and I'm going to bounce to another group.
I don't know if the fertilizer will be considered. Let me see.
Wait, what did you say, Kimberly? It cut out.
Oh, I was saying like based off like the fertilizer. I don't know like how it'll be considered either independent or dependent or even constant because it's a fertilizer. So
I think maybe constant because it'd be like the same type.
Yeah, I think it'd
Yeah.
be constant for sure. What do you guys think the independent variable is before anybody else gets in here?
I don't, well, would it be like the soil or like?
They
but I
change
don't-
the students change the soil.
Well, then they like mix the fertilizer, but then that's constant, so that's not a variable. So change the materials again. Where's their soil? Oh, what about the fast plant seed? Can they add certain amounts and then like change that?
Did
It
they?
could. Hold on, I'm gonna check.
Okay, are we all back? It looks like we are all back now. Okay, so let me ask you guys, what questions did you guys have before we get started? So we got 8 minutes just to explore your questions. What questions do you guys have about this? Like, I don't understand this or whatever. Anything. Totally open.
What exactly would we say the independent and dependent variable are? Because we said it would be independent is the trait selected and dependent is the frequency or intensity of that trait.
Yeah, absolutely. Where did you find that?
Okay.
What page?
I mean, that's just the conclusion I came to, but I suppose you could see that in like around step 4 through 7 where they actually change the variation.
Yeah, I think it's on page 4. Let's see. Yeah, so let's see. Yeah, step 6. This is you're now ready to make selection decisions. Directional selection. Remember how we were, when you select for one extreme. So in this case, they were selecting the hairiest plants, the ones that had the most trichomes. And it said that they were to find the top 10% of plants. So there's 150 plants, and I'm sharing my screen. Can you see what I'm looking at or not?
Yes, we can
No.
see PowerPoint.
Oh, wait. Oh, we see the PowerPoint,
Okay,
but
hold on.
not the
Whoa,
doc.
whoa,
Are we
whoa.
supposed to see the PowerPoint? Oh,
Oh, hold on. I thought I had the document open. Yeah, I'm looking at step 6. Sorry. So step 6 says, yeah, 10% of the plants are going to be selected. So there's 150 plants. 10% of 150 is 15. And so you select that top 10% of plants, those 15 plants that have the hairiest. Trichomes are really hairy. I'm going to show you what those are in just a second. There's those little hairs that are all over plants. So you cut those, use a scissors, you cut the tops off of all the other ones. You throw all the other 90% out. And then you just cross-pollinate those other 15. You literally mate those plants together, and then they produce seeds. So then exactly, but you're saying the dependent variable will be the frequency. So the next generation, if you breed the hairiest plants together, the next generation the frequency of hairiness should increase. And then you do it again, and then you're going to get hairier. Every generation should get hairier. It's kind of what you would expect, like if you've got a bunch of dogs, you got 100 dogs, and you breed the 5 smallest dogs, the next generation of dogs is probably going to be smaller than the previous generation. You keep doing that till finally you get a dog that's so small you can put it in a purse. That's evolution, changing allele frequency over time. Okay, what other questions you guys have? We have 5 more minutes.
Well, my group noticed this throughout the doc, but we just wanted to double check if it was intentional or not. There were like typos, but like kind of like weird typos, like in
Okay, did you
different.
did you use the Google Doc or the PDF?
Well, oh yeah, we used the PDF.
Well,
Well-
I'm using the PDF. I don't see the typo. So where did you
I
see a
mean,
typo?
sorry, the doc.
Yeah, the Google. Some people can't open a PDFs, and so I put this PDF here as well as the Google Doc, so that people who couldn't get the PDF could use the Google Doc. It's the same information, but it doesn't convert near as well. This is much easier to read. I'm just using that Google Doc also to make sure that I can take attendance.
Mm-hmm. Thank you.
All right. What other questions you guys have about the content?
What does vermiculite do?
Oh man, I wish I was in class. I could show you vermiculite. That's vermiculite right there. If vermiculite is this real absorbent material, it kind of looks goldish. If you've ever bought potting soil at the store to put plants, you'll see this really light material mixed into the soil. Sometimes it's white, sometimes it's kind of a golden color, and all it does is just hold water. It's very absorbent, and it holds water. If you've ever changed oil at your house, some people use kitty litter if they spill oil. Some people use vermiculite. All it is just this stuff that it's really light and it absorbs. It's got a ton of surface area and it absorbs up water really well. That way these plants don't dry out. And there's these really small cups, so you don't want them to desiccate or dry out really quickly. 2 more minutes. Ask questions, guys. Come on. Because I'm about to start asking you all questions.
Yeah, I have a question. Is there any need for this biology stuff? Or
Yes.
is history really the most important class?
I would
that's my
say
question.
that if you want to study something that's Eurocentric and has a bias, you should study history. If you want to study something that's objective and has no bias and can generate a lot of income and propel society forward, including how to solve pandemics.
Seems like we know a lot more stuff about our world, the way it works, our culture, the way it works with the history class and the superior history teacher. Hi, Mr. Pope.
He teaches a bunch of Eurocentric crap. Don't listen to him. All right, if you guys don't have any more questions, I'm fixing to split. I'm fixing to go. Oh, there was a question in the chat box. Yeah, let him have it. Bunch of garbage. We'll never get to Mars with that kind of with that discipline. What are you gonna do? You know. Okay, so I'm doing this for a couple reasons. One, I think that it's kind of a double whammy. You can maybe increase your ACT skills in science because you're able to disseminate information about experimental design. It's also an objective way to look at life, right? And again, objectivity means you look at a set of information and you make decisions based on facts, not on emotion. And so when we're reading that, we're just looking at this like, what was the independent variable? What did we change? What could we measure from that change? Those are not subjective questions, right? Those are straightforward, factual questions. What do I have to do to keep things the same so I know that I have an answer, so that I can trust things? I mean, we've got people that have vaccine hesitancy in this country because they think that they're going to put microchips in them from a vaccine, which is insane. We can't have that kind of thing. So we need to be thoughtful on how we approach this. Now, also, you have IB proposals, and those IB proposals, those revisions are going to be due in a little less than 2 weeks. So I think 2 weeks or a week from this Monday. So we only have 2 weekends remaining. I'll have office hours today and on Monday to talk about your proposals if you still want to talk. So first question is, does selection cause variation? We'll ask George. George, does selection cause variation?
Yes, it does.
Okay, so let me ask you a question. You've got really strong selection pressure to run fast. Somebody's throwing a ball. Can you run faster because you need to?
Yes, I'm assuming yes.
Really? You can run faster just because you need to? So like if there's a cheetah chasing you, you can run faster?
Well, oh no, then no.
Oh, no. Okay. So you don't get to change just because there's a need. So in this case, somebody drops, I don't know, they're going to drop a 500-pound bowling ball from a building. I can't generate I can't, because my head's hard. I can't just because there's a ball coming for me, make my head stronger for that reason, right? Because there's selection. I'm about to get hit with the ball. So okay, George. And so that would be true. We cannot. Selection only modifies and maintains variation. What causes variation in this case? What actually causes variation? Let's go to Dev. Dev, what causes variation? I think Dev had to leave, actually. He sent me a text. So let's go to, I want to hear somebody else. Becerra, what causes variation? I mean, yeah, what causes it? Oh, don't chat.
Competition.
Nope, it does not. Competition does not. Oh, Dev did answer the question. He put it in the chat. Dev, could you come on and just say it? It's better.
Mutations and crossing over.
Yeah, crossing over from sexual reproduction, right? Yeah, meiosis. So meiosis is going to give us sexual reproduction, is going to do it, and then mutations. No, competition doesn't cause selection. Because I'm competing with somebody doesn't mean that all of a sudden I'm going to change my traits. I cannot change my DNA, no matter how much I want to. I'm about to go into a hospital to take a stress test because I'm having some medical problems. And I can't go, oh, hold on a second. Oh, Jesus, I want to live longer. I'm going to change my DNA. I just don't get that opportunity. Okay, so what are trichomes? I need to explain what these trichomes are. So some of you guys asked me that when I went to your breakout rooms, and I didn't get to go to all breakout rooms. Trichomes are these little bitty hairy structures, these little bitty hairs. And just like the hairs on your head, trichomes on plants kind of shield you from like the wind. And wind will dry you out, right? If you guys know, if you go out and it's a windy day, your skin gets really ashy. It's because the air is going over your skin, and it's almost like the air increases its surface area, even though it doesn't. It's just the air moving across your skin is going to, as it goes by, it's going to wick out moisture. Well, the same thing with these trichomes. they are going to prevent water loss, and they also keep predators from eating them. So the people that grow marijuana, this is what they select for. Marijuana, people say, oh, look, there's little crystals on this marijuana. It's not crystals, idiot. They're trichomes. You know, they keep hitting the pipe and seeing crystals, but they're not really crystals. They're just these little hairs. And what these little hairs do is they produce toxins as well that keep animals from eating them. The animal eats it and goes, Jesus Christ, man, what the heck is this? This stuff is terrible. It makes my mouth hurt. Or it confuses them, like the compound in marijuana. You know, so they get confused, and so they don't know where they are. They forget where the plant was. So these are structures that help protect the plant. That's what trichomes are, and they're a trait. Cotyledons are these. So I wanted to select something else a little different. A cotyledon, if you've ever seen on a bean has two halves. They're these first little leaves, and they're full of carbohydrates and proteins, so that when a plant comes out of the ground, they're not these plants are not photosynthetic. They just have these little stores here, just like an egg and a yolk, that yolk and an egg, the little stores of carbohydrates and proteins, so that they can start growing. And then if you've ever grown beans, you see they kind of shrivel up and dry out after just kind of like your umbilical cord dries up, falls off. You're not attached to your mother anymore, just like that. They're analogous structures. A plant would have these to give them the first start, right? The initial growth of a seedling. And these are structures that you're going to need to understand in just a little while. Ooh, I got questions. I love it. I like questions. But guys, chat to me. Jeez. Are beans out of plants or trees? Trees are plants. I'm not sure what you're talking about.
I mean, like, cause I don't know why I thought they were like out of a tree, but like by plants, I mean like cotton is out of like a plant, like a small plant. You know what I mean?
You mean like a shrub or a vine?
Yeah.
Beans are generally vines. It depends on the species.
Okay.
Some of them are bushes, but I don't know that there's any tree that produces beans that I'm aware of. Oh, I guess Brazil nuts, but those are nuts. Yeah, I don't think so. I think they're all going to be vines or bushes.
Okay. Thank you.
Welcome, baby. All right. See if we can get a little further. And then these things are important too. These are cool. They look like little lips, don't they? They're really nice. Look at those lips. So what these stomata are for gas exchange. And remember, in photosynthesis, we have a final exam in about a month, so we should probably start to review a little bit at the same time. The gas that goes into it is going to be CO2. The gas that comes out of it is going to be O2. So you need gas exchange. They'll also use some of that oxygen for cellular respiration as well. Remember, they have mitochondria. But the problem with this is an evolutionary trade-off. When they need gas to get in, they need CO2, but they lose water at the same time. So this is definitely a trade-off. It's not a perfect structure for plants, but it works well, semi-well. But these are also a trait. And you'll see more about these traits on Tuesday, which is something my students discovered, which was totally blew my mind. But we're measuring, this will be another trait that the students decided to measure. And I've got their data, and we're going to play with their data on Tuesday. And you guys are going to have a pretty tight assignment on Tuesday, so really help me out here. So what do you think would be the independent variable? So somebody other than Keaton, what was the independent variable? And I have to be in this hospital in 22 minutes, so I need you guys to hustle. I'm ignoring the chat. I don't want to see the chat, guys. Talk. We're not going to get there by chatting our way through this.
Was it the frequency of the variation?
The frequency of what again?
The variation or no?
I don't know about the frequency. The frequency, I think, was over here, maybe dependent variable. The independent variable, what was it? Look, you can look on that PDF. I think it was page 4, step 6. What were they doing? What is the experimenter change? The independent variable is what the experimenter does that causes a change.
Is it artificial selection?
Okay, it is. And what specifically were they selecting?
The hairiest plants.
Yes, and what they selected, and by the way, so let's see, there is Chastain. Chastain, you're in the room with my students. Can you hear me? Chastain, can you hear me?
He said yes.
Okay, ask him to, okay, no, ask him. Hey, you need to, please make sure the students are writing this down, this chart. It's an IVCDV chart. They need to be writing this down. Anytime you set up an experiment, always start here for your procedures. Know what you're doing before you start to do anything else. This is just like a basic way to do this. Start with your independent variable and then we'll move forward. So selection of the hairiest plants. And in this case, it was a top 10%. Okay, let's see. That's what somebody said. Let's see if that's right. Yes. And it was trichome density. Good job. Who was that said that, by the way?
Me.
Sarah. Good job, Sarah. Sarah's the boss. Sarah Ita is boss. Okay. What are my constants? What are some constants? We got to have at least 4. We can come up with more. Let's start brainstorming.
Fertilizer.
I'm sorry?
The fertilizer.
Okay, so fertilizer is consistent, right? Okay, so let's see. Fertilizer is consistent. You want to have
the soil?
Soil is consistent. What was the soil? I forgot. I honestly forgot. I think it was Jiffy, right?
Yes.
Yes, Jiffy. I've got some in my classroom. So a lot of these things, remember, I have some of this stuff in my class for you guys to use next year. What else?
Type of plant seeds?
Yeah, the same species of plants. It was Brassica, Rapa. And I plant this, tons of this every year. I plant this as a cover crop for my, where my deer are, where I hunt.
Constant would be the wicking twine, right?
Yeah,
Wicking twine.
Yeah, wicking twine. Wicking twine or string. All right, and that was to pull up water, right? So that was like the little cups, and they had a little water, little string that came out, and so you poured water in here, then the water would travel up the wicking twine. What else? Think about variables for photosynthesis. These are plants. What would you?
Light?
Yeah, light. So light intensity. What else about light? Light intensity and what else? Because this is a common problem I see in your guys' eyes. Light intensity and what else?
Water.
No, stick with light. So light intensity has to be constant. What else has to be constant about light?
The color.
Okay, so the spectrum or wavelength, absolutely. So wavelength would need to be the same as well. And there's something else, and I'm just abbreviating here. Wavelength. What else?
Angle?
So the angle of the light would need to be the same. So if you had a light, so this is the light, it's on a, and then these are my plants here. You'd want to make sure the light's coming directly at it, so the light's coming directly towards it. If not, they all need to be at another, if you had them coming from the side, they would all need to be from the side. However you would set that up. There's still something else that matters.
The amount of time?
Yes, the time, the photo period. And I'm actually going to write that, photo period. Photo period is really important in nature for a lot of reasons. Like animals will start to have breeding seasons based on photo period. One of the reasons for human depression, they think, is because people stay up late and they get more light than they should, and they're not getting the appropriate amount of darkness. The photo period in your eyes is being changed by the artificial environment of computers. Shut the dadgum computer off at night. So plants also respond by seeding according to photo period. You get lengthening days. This plant's go, oh my God, it's springtime. Let's make flowers. They don't want to do it when it's the way days start to get shorter. Usually plants in the fall say, well, it's not the time to breed. What else? Let's try to get one more thing. What about water?
Type of water?
Yeah, maybe the type of water, the volume of water. There's a lot of things. We're just brainstorming here. Yeah, there you go. These were just my initial thoughts whenever I was putting this together. Now, one of the things also we want to keep consistent is what part of the plant are we going to actually analyze? So you would need to select, in this case, if I'm selecting trichomes, I need to select the same leaf on the same branch, right? I don't want to check for the amount of hair on a person's skin based on where, different parts of the body. So if I analyze my upper lip, obviously I'm going to have more hair than if I analyze my forehead, right? So the same place that you're measuring on the plant is going to matter as well. So we got some chats here. Thank you, Randy. I just saw that. So what is the dependent variable? Where's our dependent variable? What's our dependent variable?
The frequency
Frequency of what?
of the
Actually, I'm going to change that to density. Of what?
Is it the density of the trichomes?
Of the trichomes. Now, you remember those other structures? So let's go back and look really quick. The stomatas and the collodons. My students decided that they were going to take what they did for their experiment. They said, well, shoot, I remember we did this. It takes 3 months to do this, by the way, literally 3 months. They grew these plants. They selected the top 10%, cut the other 90% away, and literally crossed them acted like little bees, had these little wands that look like mascara applicators, and literally took pollen from different plants and crossed them, took the seeds from those, raised another generation, the F1s, and then did the same thing and got F2s. It was really cool. But they wanted to know, because remember there's gene linkage. They wanted to know about gene linkage because they remember gene linkage being something that kicked their butt in genetics. Well, they want to know is there a positive correlation? So if I select trichomes, if trichomes density increases, will also collodons, will they also increase? Are these genes linked? And they also did the same thing for stomata. Will stomata increase? They're like, do these things are these genes for these traits, are they right next to each other on the same chromosome? That will be a cool independent. So some of you guys are measuring more than one dependent variable. Some of you guys are doing this in groups. And so you need a different dependent variable. So my students that did this in a group decided to do 2 dependent variables for their IA. And this is literally somebody's IA. I'm breaking it down for you guys. So you can see, how do I design this? Always kind of start with a chart like this. Now, obviously, there's a bunch more constants. There's a lot more constants. And I'm not putting any assumptions up here. So my next question is, what's their control group? This is the thing that most of you guys are leaving off that's driving me crazy. What's the control group? Because you have to identify the control group. I got to compare this to something. Science is nothing. How do I know the vaccine works if I compare vaccinated people against unvaccinated people? Who gets sick the most, right? Well, that's how we know if a vaccine's working. So far, we've had 0 people that have been vaccinated with this vaccine, any of the vaccines, get COVID, go to the hospital and die. There have been people that have picked up COVID, but it has been so mild a case that they didn't even know they were sick. But people that are unvaccinated are still getting COVID. And some of them obviously are dying. Most of them are not. But what's our control group? All right, what time is it? I have to be in the doctor in 10 minutes. I'm gonna drop you guys back into your breakout rooms. And you have 2 minutes to tell me what the heck is the control group. Go for it. Find the control group, yo. Find the control group. Randy, Diego, you guys can all stay in your room. Just talk.
Okay, Brianna, what do you think?
Brianna, what do you think, kid? Hello, Brianna. We're talking to you.
I'm like rereading real quick. Give me a second.
It's not going to tell you what the control group is. You have to think about it. It will not tell you. You just have to think.
Well, we have those fast plant seeds, which are, I think they said optional in the thing.
Yeah, you could use, those are just the species you're using. That's a constant. That's a control variable. It can't be your control group.
Okay, just to get a clear understanding, like what's the main difference between control variable and group? So
a controlled variable is something that you keep the same in the experiment, right? So like if I wanted, like let's use the COVID vaccine as an example. When you're looking at infection rates between people that have been vaccinated against those that haven't been vaccinated, you also measure by demographics. So how are people that are female doing compared to other females? So one of the constants that you would keep, you'd want to look at age class as the same, the sex is the same, the race is the same. Those are the things, the conditions in the experiment that do not change, They're constants, they're controlled variables. So not things that you don't change. It's not what you compare to. I'm not comparing the volume of water to the density of trichomes, but I do have to keep the volume of water the same, because if I treat some of them with more water than others, maybe the volume of water causes trichomes. I don't know. I'm nervous about going in here and getting this test in a minute. Not real happy about it. Yeah, the control group is what you compare the results to. Absolutely. Let's see if they figured it out. I've got blood pressure issues and I got diagnosed with a slight heart condition. It's no big deal. It's not something that's too terrible. I just need to make sure I take care of it. Thanks for asking, Sarah. Okie doke. Anybody have any ideas? What's our control group, yo?
Jacob said that the control group was one of the plants that wasn't mutated, or
I don't know that we didn't measure mutations.
Or that wasn't
Here,
crossbred?
wait, I got it. Yeah,
Okay.
so it's basically a plant that wasn't like bred to grow more hair. It was just
Yeah,
like a
exactly.
regular.
Who is this that says this stuff, Jacob?
Yeah, it's me, Jacob.
Okay, so you're right. So remember, our independent variable is selection, right? So which plants were not under selection? Remember, we had generations. We had the P generation that led us to our F1 generation, which led us to our F2 generation. Which plants were not under selection?
The 90%.
That's a good idea, but just that's a really good idea. But they were technically under selection, but whose parents? So were there parents that led to these right here, the P generation? Did these parents? Well, these are our seeds. We use seeds to get these. Did these plants have parents? Do you think?
Yes.
Yes, they had to have, unless they sprang out of the ground, right? Which is not possible. Yeah, so these seeds, they did have parents. So since their parents weren't under selection, this was our control. This was our control, the P generation. Trichomes were not under selection prior to the P generation. So that is the control group. It's not going to tell you exactly. I want you guys to think about these things rather than just being told what the answer is. Really think about it. So everybody should have this written down. I hope that you were taking notes with this. We're going to go through and analyze data. I'm literally going to walk you through, and this is a great experiment to use because A, it helps us with evolution, and B, it helps you with thinking about how you're going to design and use your revisions. If I gave you a ton of comments about your independent variable, your dependent variable, and your controlled variables or constants, make a chart, have a thought experiment. Think about what would I have to keep the same for this to have an effect. Really think about everything comprehensively so that you can be objective and not have a bias. That's what's so cool about science, is it's completely unbiased, it's completely objective, but you have to work to meet that that level of expertise in learning how to alleviate bias and subjectivity by setting it down and thinking about what would my constants or controlled variables be so that my independent variable can actually potentially have an effect. You don't know if it's going to have an effect, but it could. And if you have a bunch of other extraneous variables, well, then you don't know. And that's what sucks about history, Chastain. There's so many other variables. How do you know one thing was a cause of something? You don't know. It's a bunch of subjective, mumbo jumbo. Where are you, Chastain? All right, guys, I do have to walk into this hospital, so you're going to get cut loose 10 minutes early. I will see you guys on campus in about an hour and a half, 2 hours at the most. You guys be good.
Bye.
Oh,
Goodbye.
there's some questions. I'll answer your questions.
Bye.
Yeah, you got it, Sarah. You got it. Thank you. Thank you, Randy. Thank you, guys. You guys be good. I'll make this PowerPoint available